Hello everyone. Welcome back to Dr. Lou's VCE Chemistry channel. Today, let's learn together about the 2023 chemistry examination. Now, we analyze the biochemistry sections. My explanations may be flawed. If any teachers, parents, or classmates find errors, please feel free to provide your guidance. The pathway cannot change the status of thermochemistry. The combustion of glucose is exothermic in vitro, also respiration of glucose is exothermic in vivo. So, B is correct. All molecules are triglycerides and have the same number of carbon atoms. So, their melting points are dependent on the number of carbon-carbon double bonds. The more carbon-carbon double bonds, the lower the melting points. There are 3 carbon-carbon bonds in Z, 1 in X and no in Y. So, from highest to lowest, the order of melting points is Y, X, and Z. All 3 statements are properties of coenzymes. Coenzymes assist enzymes to catalyze a reaction by binding loosely to the active site of the enzymes and supporting the transfer of electrons and atoms during the reaction. Omega-6 fatty acids contain a C equals C double bond. This creates a kink in the chains that prevents them from packing as close together as the chains in saturated fatty acids. So, omega-6 fatty acids have lower densities and melting points than saturated fatty acids. So, A is correct and B is eliminated. Unsaturated fatty acids have fewer hydrogen atoms than saturated fatty acids. So, C is eliminated. Unsaturated fatty acids have fewer hydrogen atoms than saturated fatty acids, so omega-6 fatty acids undergo substitution reactions less readily than saturated fatty acids. So, D is eliminated. Denaturation can also be caused by certain chemicals as well as changes in pH or temperature. So, A is eliminated. Sometimes, denaturation causes the irreversible change of a protein structure. So, B is eliminated. Denaturation changes the 3 dimensions of protein and does not affect the primary structure. So, C is eliminated. Denaturation alters the secondary and tertiary and quaternary structures. So, D is correct. Linoleic acid is an omega-6 fatty acid, and other fatty acids are not omega-3 fatty acids. So, 0% is the correct answer if the name linoleic acid is used. However, all students are awarded 1 mark because of conflicting information. I suppose that the last raw could be linolenic acid but print as linoleic acid. Apply syllogism to answer this question. Saturated fatty acid does not undergo oxidative rancidity, and coconut oil contains a high percentage of saturated fatty acid, so coconut oil is resistant to oxidative rancidity. Oil is triglyceride but not fatty acids. In the body, coconut oil is hydrolyzed to fatty acid and glycerol. So, the answer to the question C is glycerol. Coconut oil is a triglyceride. 1 glycerol and 3 caprylic acids condense this triglyceride, and 3 water molecules are eliminated. We list the carbons, hydrogens, and oxygens of reactants and products, and calculate the number of them step by step. Finally, we get the number of carbon is 27, hydrogen 50, and oxygen 6. Explain why vitamin E dissolves in coconut oil. According to the principle of likes dissolve likes, a molecule can dissolve into another molecule if they have a similar intermolecular force. Both coconut oil and vitamin E are nonpolar molecules and dispersion forces are a main intermolecular force. When vitamin E is in coconut oil, the dispersion forces would be formed between vitamin E and coconut oil. So, vitamin E can dissolve in coconut oil. This question belongs to electrochemistry. Ask for the half equation in the cathode of the coconut oil fuel cell. Generally, oxygen gas is the final electron acceptor in fuel cells. The cathode is where oxygen gas gains electrons. There are 2 equations that oxygen gas gains electrons, one is in an acidic electrolyte and the other an alkaline. This fuel cell is running in an alkaline electrolyte, so the equation in the last row is selected. Don't use a double arrow in the answer because reduction is one way in the cathode. The electrode in the cathode should be made of PT as a catalyst and porous to increase surface area. Compared with direct combustion, the fuel cell has higher efficiency because the fuel cell directly transforms chemical energy into electrical energy. Give one reason why coconut oil may not be a practical fuel for a fuel cell, but this reason is not referred to as environmental issues or sustainability. This question is related to the disadvantages of fuel cells. One of the disadvantages is okay but should be linked to coconut oil. For example, coconut oil is viscous or not soluble in water. Other answers are acceptable. For example, the output of coconut oil is not large, coconut oil cannot be stored for a longer time, and coconut trees only grow in tropical areas. The two monosaccharides are joined by an oxygen atom. The connection between the monosaccharide units is called an ether link, which is called a glycosidic link in carbohydrates. 2 marks cover 2 points. The glycemic index is a measure of the effects of food on blood sugar levels. Glucose has a glycemic index, GI, of 100. After hydrolyzing lactose, glucose is produced and galactose will be converted to glucose in further metabolization. So, the breakdown of lactose contributes to the GI of lactose. The key word is essential. Proline can be synthesized in the body, so it is not an essential amino acid. In an acidic solution, hydrogen ions are in excess. So, carboxylic groups absorb hydrogen ions, and amino groups also absorb hydrogen ions. After absorbing hydrogen ions, carboxylic groups are neutral, and amino groups are positive charges. The positive signs must be labeled on nitrogen atoms. State 2 reactions about proline in the body. Protein in food is hydrolyzed in the digestive system to produce proline, and prolines condensate with another amino acid to synthesize protein. The alpha carbon atom of proline is chiral carbon, and this chiral carbon has 2 enantiomers. So, a proline has 2 optical isomers. The incorrect optical isomer of proline would alter the primary structure of the enzyme and further change the shape of active sites of lactase. When the shape of the active site is changed, this active site cannot match the shape of lactose and cannot catalyze the hydrolysis of lactose. So, only one of proline's optical isomers is found in lactase. This experiment is to compare the energy content of dry broad beans and dry soybeans. Colorimeters are used to measure the energy of dry broad beans and dry soybeans. After crushing dry beans, the surface area of the beans is increased, and subsequently, the combustion rate is increased. To compare the energy content of dry broad and dry beans, their energy content must be measured, so, the dependent variable is energy content. There is a heat hazard in step 4. Using a heat-proof mat reduces the chance of the laboratory catching fire. Another step is step 8. This risk is scalding or burns. Waiting for water to cool below 30 degrees reduces the chance of scalding or burns. Step by step to calculate the energy content, temperature change, the mass of water, the energy change in the water, and energy content. The density of water is not 1 but is 0.997. The final answer is 2 significant figures because the significant figure of delta temperature is 2. 2 steps to calculate the heat of combustion in kJ per gram, the energy content per 100 grams and converting this into kJ per gram. A significant number of students incorrectly quoted the energy content as being 1,816 kilojoules per gram, not realizing that the mass is given where the average quantity per 100 grams. The final answer is 2 significant figures because the data just have 2 significant figures. The experimental result is higher than the expected results. The mass of water in colorimeters is less than 300.0 milliliters, which induces the temperature change to a larger than expected value. So, the energy change is larger than the expected value. Also, the water in colorimeters is not completely mixture, which induces the temperature to rise too high on location. It is okay that any reason is related to temperature change too high. Suggest one improvement to the experiment method and explain. An improvement could be to ensure the accurate volume of water. This would allow an accurate delta temperature and hence a final energy content closer to the true value. Or, an improvement could be to stir or mix the water in the colorimeter completely. This would distribute heat energy evenly and hence give an accurate delta temperature. So, the final energy would be closer to the true value. The energy content of soybeans is 18 kilojoules per gram, and that of broad beans is 8 kilojoules per gram. So, the energy content of the soybeans was higher than the energy content of the broad beans. Also, it is an acceptable answer that no valid conclusion can be drawn from this experiment due to the experimental error that occurred with the soybean trial. There are 5 multiple-choice questions worth 1 point each, totaling 5 marks. There are also 3 short answer questions worth 39 marks in biochemistry. The overall score for this section is 44 points. With a total possible score of 120 points, the percentage for this section is 36.7% of the total score. Thermochemistry is 22.5%, electrochemistry is 19.2%, organic chemistry is 21.7%, and biochemistry is 36.6%. Thermochemistry covers 5 chapters, 1, 2, 3, 7, and 8. Electrochemistry covers 4 chapters, 4, 5, 6, and 9. Organic chemistry covers 5 chapters, 10 to 14. Biochemistry covers 3 chapters, 15, 16, and 17. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Lou's VCE Chemistry channel.